spontaneously re-anneal and form double-stranded helices again. Instead of heat, sometimes we can use alkali, sodium hydroxide, and then if we neutralize to reverse that insult, the DNA strands will come back together again. So it's just a very convenient property of DNA based upon the hydrogen bonding interactions that hold the double-stranded molecule together but can be exploited in this way. The simplest way we can use that property to our advantage is to design a probe, a short single-stranded DNA. Sometimes it's several hundred bases. In this little diagram, it's just a few bases. We make that single-stranded DNA a probe by adding some kind of a chemical label to it. That label might be a radioactive phosphate that we can add to the 5' end, and a lot of classic experiments were done that way. These days we have much better access to glow-in-the-dark molecules, things that emit light, and they're much safer than radiation, and so a label in some fashion added somewhere to the single-stranded probe allows you to find that probe again wherever it has gone. The general idea is that probe will stick to a single-stranded target sequence, oftentimes very, very much more complex than the one shown here. But students certainly should know that the probe will stick to a place which is both complementary and anti-parallel. And so students are sometimes asked to indicate where a probe might stick, just as a test question kind of thing. So the backbones must be anti-parallel, and students will often overlook that. The simplest technique that you can do with this we saw last time. When the probe is radioactive, we will often call the experiment autoradiography. If you put a piece of X-ray film down on top of your experiment, wherever radiation is being emitted by the probe, the X-ray film will be blackened. These days with fluorescence probes, we call it fluorescence in situ hybridization. In situ refers to chromosomes as they are. So take the chromosomes out of a cell, but don't change them chemically in any way. Leave the DNA strands that make up the chromosome organized around a protein scaffold so that they still do look like chromosomes, colored bodies like this. This is a diagram of two homologs sitting side by side. These are condensed chromosomes. You should already know that chromosomes are only condensed as they are getting ready for a nuclear division. That always happens after DNA replication. And so these duplicated chromosomes are really two double helices held together by a centromere. So this is one homolog, two sister chromatids, and this is a similar homolog, same chromosome, same size, same shape, maybe different alleles, but all the same genes. If these duplicated metaphase chromosomes are stuck to a glass slide, you dip the glass slide into sodium hydroxide, and you can make those double-stranded DNAs locally separate into single strands but remain adhering to the glass slide. And then you can wash over that your single-stranded probe, and in this example the yellow glowing probe does stick to its anti-parallel complement. It doesn't stick to both strands, it's identical to one strand, it's complementary to the other. But since these two sister chromatids are identical, that's why we see signal on 2 spots on this duplicated chromosome, 4 spots in this metaphase spread, because there are 4 chromatids at this point during cell division. So we're not talking about cell division in this class, but that's why it looks the way it does there. So the ability to use a probe to find a complementary antiparallel sequence, in this case in metaphase chromosomes. The other place you can use a probe to find a particular DNA sequence is in an electrophoresis gel like this. So in this diagram from your book, we imagine that we have taken human or mouse, some genomic DNA, 3 times 10 to the 9th. It says here we've cut it with a restriction endonuclease, maybe like EcoR1. We calculated last time that that will generate almost 1 million fragments of all different sizes that range from long fragments all the way down to very small fragments. We can't find any individual fragments because there are 750,000 of them, and it just looks like one big smear. Max knows gels are very fragile. And so you don't want to do this experiment in the gel. We simply want to transfer the DNA out of the gel onto the rigid paper support. And so nitrocellulose paper was what was originally used for this kind of an experiment. These days we will rather use nylon. A nylon paper support is much more durable, will last for decades as far as I can tell. And this is what you may have heard referred to as a southern blot. Southern is actually somebody's last name who invented this procedure. The blot is what we're seeing here. The DNA is in the agarose gel after electrophoresis, all spread out according to its length. But to get it out of the fragile agarose, we are just using a Pampers diaper, basically. Bounty paper towels, real low tech. We're just sucking moisture up from below the gel. There's the gel in gray. Up through the nylon or nitrocellulose membrane shown in green, and then the moisture from below going up through the gel goes into the paper toweling. As it makes that short trip, it should drag the DNA out of the gel, and the DNA will bump into the nitrocellulose or nylon filter. Then you can simply peel the filter off the gel. DNA's out of the gel now, and it's all covalently bonded to the filter, so it's there for good. If you notice right here, the solution we use to do the blotting is an alkali solution, and so that DNA affixed now to the membrane should be single-stranded and a perfectly good target for probing. So on this nice durable membrane now, you stick that into a sealamine bag, throw in some radioactive or fluorescently labeled single-stranded probe, and that probe will now stick to its complementary anti-parallel sequence that it finds somewhere in that array of electrophoresed restricted DNAs. Lay your X-ray film down on top of your experiment and you can blacken the locations where the probe is now located. So that's all a southern blot is getting DNA, restricted DNA, out of a gel and onto a solid support. What you can do with this filter paper now, after you've exposed your X-ray film, you can put this in boiling water, knock all the probe off, stick it in a drawer, take it out 5 years later, probe it with something else. And so once you've made your southern blot, you can probe it with any number of things that you might be curious about. In this example of a southern blot here, what they were curious about was the beta globin gene in this particular family. This couple has produced a child who has the symptoms of sickle cell anemia, and so presumably the child is homozygous for a defect in the beta globin gene. Presumably these parents are heterozygous for that defect because they don't have the disease symptoms themselves, but they must be carriers if they've produced a homozygous child. Usually this experiment is run to figure out what the other normal siblings might have in terms of their beta globin gene. Do they have nothing but normal wild type copies of the gene and therefore will never pass this on to their kids? Or are they themselves carriers? That's often the kind of thing that a carrier might want to know. Or any phenotypically normal person might wonder if they are a carrier, especially around the time they are planning to have a family of their own. So this kind of experiment is a little bit old-fashioned at this point, but there's a lot of things that we've learned that we can see here. Here they're showing the normal adult beta globin gene, not very much of it, just some of it. They're showing this tiny little section of it, which includes a MISTI2 target site. So just from the way that's written, you can tell that's the name of a restriction endonuclease. This happens to be the target sequence for MISTI2. CCT, and then any base in the middle, AGG. That is a palindromic sequence. That ought to behave just like any nuclease we've seen that has a 6-base target sequence, because that 7th base can be any of the 4, and so N is sometimes used to stand for any of the 4 bases. They're telling us here that the sickle cell gene is different from the normal gene by an alteration in just one base. So where there should be an A, in the sickle cell allele there is instead a T. That T changes this sequence so it is no longer a MISTI2 target site. in addition to being a single base change that also causes the beta globin protein to be mostly okay, but it changes its shape inappropriately under conditions of strenuous exercise, and in your muscles the pH drops and the beta globin protein tends to kind of fall apart and disrupt the shape of the red blood cells. And I think you probably know all of that symptomology because beta globin is an example of almost everything in almost every course you've ever taken and ever will take in college. But as an example of the southern blot here, we can imagine a person who is phenotypically normal. On both of their homologs, they have exactly this spelling. Take their genomic DNA, cut it with MSD2. There'll be a target site to the left of the gene. There'll be a target site to the right of the gene. There will also be a target site in the middle of the gene. If that homozygous normal person is now probed with a sequence of DNA that covers this extent of the gene, that probe will want to stick to this fragment here. The probe will also have homology and will want to stick to this fragment here. And so the normal individual will show two bands of hybridization, and they will be on the smallish side. And so this normal individual here, who has only this chromosome, will show only this signal on the southern blot. By contrast, let's consider the sick boy who has only this sequence. MSD2 will cut his DNA to the left of beta globin, to the right of beta globin, and that's it. We've lost the MSD2 site that is within the gene, and so this probe will now stick to one fragment, which is the size of the small fragment and the small fragment here added together. And so only a single band of hybridization is seen for this homozygous individual who has only two chromosomes that have this top sequence as shown. A heterozygote will have one chromosome like this, one chromosome like this. The normal chromosome will give us the two bands. The mutated chromosome will give us the one larger band. So we can almost indicate that this signal here is the wild type allele of beta globin. This signal up here is the mutant sickle cell allele of beta globin. And so everybody reveals on this southern blot their full genotype for that particular site in the genome. And so we said, when we look at human genomic DNA, 3 times 10 to the 9th base pairs, we need a technique like this, and we need a probe so that we can find the one sequence that we want to study. So we're ignoring all of the DNA that's there. We are seeing only the location where the probe will stick. This is a little bit old-fashioned. This technology has been used for 25 years. It's been replaced with cheaper and safer and easier stuff. But importantly, that new stuff, the result looks just like this. So it is good for us to learn how to interpret even this old-fashioned kind of an experiment. So this one I think I made up, I think for a test question. So this is the kind of thing, we'll take the time to read this. And this question was designed to resemble the sickle cell southern blot that everyone does get taught. The following map of the phenylalanine hydroxylase gene shows the positions of 3 EcoR1 sites in the normal allele with the distances shown in between in kilobase pairs. The R1 site marked with a star is destroyed by a point mutation that also leads to a loss of function recessive defect that causes phenylketonuria. And PKU is probably a disorder that all of you should know. You all know sickle cell anemia because it's such an important example in your undergraduate curriculum. So is PKU. If you drinking a Diet Coke, you look at the Diet Coke can and it says don't drink this if you have PKU, and maybe you don't have PKU and you've never noticed that. But NutraSweet is 3 phenylalanines in a row. If you have phenylketonuria, you have this defect. You can't make phenylalanine hydroxylase enzyme, and you can't clear from your bloodstream excess levels of phenylalanine. And so the way you avoid symptoms as an individual with PKU is restrict the phenylalanine in your diet. So phenylalanine certainly is an amino acid. So you just have to be careful what you eat so that you never build up high levels of phenylalanine. If you do, that's not consistent with normal brain development. That's why every child in the world now, on the day they're born, gets blood taken from the bottom of their foot, and they test to see what's the level of phenylalanine in that blood. If it's high, they get the restricted diet, and they stick to this restricted diet. They don't develop severe mental retardation. And they stay on the diet until they turn 16 and get their driver's license, and every one of them drives straight to McDonald's because they've heard about this wonderful food. And luckily their brains are mostly developed fine at that point, and they don't do themselves too much harm. So this is the kind of a case where can we detect people's genotypes? It's a recessive mutation. Maybe if you're a carrier, you'd like to know that before you marry another carrier and have a chance at producing 25% affected kids. Not a problem really, because there's certainly this treatment that works beautifully well, but it was an excuse to come up with a southern blot for you to think about. Imagine that 3 individuals, normal homozygote, heterozygote, and a mutant homozygote, so we should get all 3 genotypes on this gel. Each provide a DNA sample. Samples digested with R1, so you're picturing 750,000 fragments, all different sizes, lined up from top to bottom. These digests are electrophoresed with a size standard that consists of a set of DNAs that measure 1, 2, 3, 4, and 5 kb. So that's an important part of a test question. I've got to find out that each and every one of you knows where the 5 kb band goes and where the 1 kb band goes on this diagram. Okay, and about 1 in 3 of you, so look to your left, look to your right, 1 in 3 of you won't get it right. And so this is where you have to find that out early in the course. Big, long DNAs are the ones that move slowly. Short DNAs can squiggle through the gel much more quickly. So that's certainly something that we need to assess on your first exam. Here's the rest of the question. If the sequence indicated by the red box is used as a probe, what bands do you expect to appear on the gel? Draw them. The normal homozygote, if you understand the story that we've read, will have all 3 EcoR1 sites. It looks like this probe will stick only to the 3.5 kilobase pair fragment. Remember, there's 750,000 fragments going off to the right, going off to the left. This is the only one that we will see because we're only going to see the one that the probe sticks to. And again, showing me that you know how electrophoresis works, you can put that 3.5 kilobase band right there. Being a homozygote, that's the only chromosome he's got, and that's the whole signal. The heterozygote will certainly show you that, and in addition, we'll have a chromosome that lacks this EcoR1 site. That means the probe is going to stick to this overall fragment, which you can easily add up, because we talked about addition last time, to 5. And that's where that would belong. Okay, and an individual who had only the mutant chromosome would lack the EcoR1 site in the middle and would only show the 5 kilobase pair signal there. Okay, that only differs in one small way from the southern blot example that we saw previously. And this is how it differs. I could change the question so that the probe, instead of being there, actually overlaps the mutation site, as it did in the southern blot example. That's why in the southern blot example, the normal homozygote with all 3 cut sites, the probe will stick to the small piece because it overlaps it. The probe will also stick to the larger piece because it overlaps it. That's why we see 2 signals. That does not mean this guy is heterozygous for anything. Both of these bands are consistent with the wild type allele only. And they frequently will ask this question to see if the fact that there are 2 bands confuses you. The heterozygote will have that allele as well, and in addition, on the mutant chromosome, which is missing that EcoR1 site, the probe will stick to the overall 5 kilobase band. So 3 bands, like we saw on the sickle cell gel. This is one allele up here, and this doublet is the other allele. And the mutant homozygote, again, will have just the one allele. So again, comparing these 2 experiments, when the probe does not overlap the polymorphic cut site, you see 1 band for each allele, 2 bands in the heterozygote. When the probe overlaps the polymorphic cut site, then we see a doublet for one allele and a single band for the other allele. What typically happens to a group of students like you is you are only taught this diagram, and then this diagram is the one that's on your test. That's what's typically done to you. Show you the simple way, and they expect you to figure out the more complicated picture on your own. We've done the complicated way a couple of times, and I didn't want you to be unaware of the simple way. Okay, here's another way of doing a southern blot. So that last way was taking advantage of what we called a polymorphic target site. And this frequently happens when a DNA change that causes a disease, very frequently it will also alter a palindromic target site. Gosh, I think in the 10 times in my career I've looked for a point mutation, I think 9 of those 10 times that disruption has also disrupted a easily screened endonuclease target site. Back in the days of O.J. Simpson, who no one remembers, Marcia Clark talked a lot about a restriction fragment length polymorphism. And so that's what southern blots are good at showing us. Restriction fragment length polymorphism just means 2 different ways that something can look. This is a different way of doing it. We're not requiring a restriction fragment length polymorphism in this case. So this new approach would work even if the disease-causing mutation did not happen to disrupt an endonuclease target site. In this case, there are 2 probes. I'm using sickle cell again as an example. This would be the precise spelling of the normal allele. This is the precise spelling of the mutant allele. And under very stringent conditions of hybridization, you might require perfect complementarity in order for the probe to stick, even though it's just a 1 base difference. That means the temperature is very finely controlled, or it could mean that the salt conditions are finely controlled, so that the target has to be precisely correct in order for the probe to stick. In that case, again, we can imagine our situation where we have all 3 genotypes. We have a blot that's produced like this. In this case, it doesn't matter what enzyme we use. We're not requiring that an enzyme give us a restriction fragment length polymorphism. Instead, we're going to probe this blot with probe number 1, and maybe it will stick exactly like that, telling us that each of these individuals has the A sequence, the normal HBA allele. We'll take that blot, we'll boil it to knock the probe off, and then we'll probe it again with the blue probe, and we'll see hybridization in this pattern. And taking that information together, we can see that the person in the first lane has only A, the person in the second lane has A and S, the person in the third lane has only S, and we can work out their genotypes like that. Now, if it doesn't matter what enzyme we use in this experiment, that depends entirely on perfect homology with the probe, we can make it even simpler. We can eliminate the blot, we can eliminate the digest with anything, and we can simply take DNA from these 3 individuals and just put a drop of it on some kind of nylon paper or nitrocellulose paper. Then we probe that dot of DNA, and maybe this probe will stick, boil it off, probe it again with probe number 2, and maybe it sticks there. And again, taking the information together, we can figure out everybody's genotype. So rather than a southern blot, this is cheaper and easier and faster, and this is often called a dot blot. And all it requires is that you fiddle around in the laboratory and discover exactly the conditions that require perfect homology to allow your probe to stick. And it's a lot of fiddling. You can imagine the difference between this probe and this probe is 1 base, 2 hydrogen bonds. And so our whole experimental conditions depend upon those 2 hydrogen bonds. So it's a very delicate kind of a system. So those are some of the techniques that you can do simply knowing about this physical property of DNA that if you denature it, you can rehybridize, reanneal. You can probe single-stranded DNA and see double-stranded helices forming again spontaneously. All right, now we're done with DNA structure and DNA's physical properties. We're going to move on to a lengthier discussion of the central dogma. DNA can be used to copy other DNA by the process of replication. DNA can be transcribed into RNA. RNA can be translated into protein. And so the goal of this first third of the course is to talk about replication, transcription, and translation. And so we're finally at the part of the course where we really begin what we're supposed to be talking about. Now, I'll tell you up front, the way this material gets tested all over the world, this is not just me, it's all over the world, they assume that every undergraduate hears about replication, transcription, and translation, as I'm sure you've all already heard about. A lot of you heard about it in high school. The test questions assume you've been taught this stuff, but they want to find out, have you mixed up all these terms? There are very specific terms that are used to describe replication, distinct terms used to describe transcription, distinct terms used to describe translation. And if we mix them all up and use them randomly, you will flunk the GRE and the MCAT and the DAT and all those exams that you might want to take someday. So keeping this vocabulary straight is really what an undergraduate is supposed to be accomplishing in a course like this. So DNA replication is, there are 4 key terms. First of all, we say that DNA replication is semi-conservative. What that means is that if this is our starting DNA, 2 strands held together by hydrogen bonding, after replication, we still have those same 2 strands of DNA that we started with. So it's conserved in terms of the molecules that pre-existed and the ones we end up with. We haven't lost anything. But here they were hydrogen bonded with each other, and here they're hydrogen bonded with somebody new. And in fact, here's your 2 sister chromatids held together with a centromere. But in terms of the pairing relationship, this is a new arrangement. And so semi-conservative, both conservative and not conservative, that's what that term really means. Now, naturally, in a day, when we talk about transcription, it will be fully conservative. And so, again, completely different words to describe this aspect of these 2 different processes. So we'll make sure that you don't mix those terms up. On this diagram from the book, you can see the semi-conservative style of DNA replication as we know it. We've got the pre-existing yellow strands through the process of replication, which we'll see in more detail in a minute. They still exist, but now they are, after replication, paired with somebody new. Again, the yellow and the red strands in another round of replication are not lost. They are conserved, but now they have a new pairing relationship with the brand new green strands. And so this semi-conservative pattern, there certainly could be questions designed to see if students know what that term means, semi-conservative. In addition, we often say that DNA replication is bidirectional. We can often describe that by thinking about double-stranded DNA. I guess I can have a simple diagram like that. And if replication starts to happen, what we can observe, even if we look at an electron micrograph, we can see this happening. The 2 strands of the double helix will locally and temporarily separate to become single strands. It doesn't last very long, and almost immediately that new strand is built in. So a piece of vocabulary here, the place where replication starts is called the origin. In fact, we said the other day in lab that every plasmid has an ori. Every plasmid has a replication origin. And so the term origin is only ever used to mean the place where DNA replication begins. If we looked at this chromosome. A few minutes later, it would look like this. And that replication bubble would appear to have grown. Of course, it's not going to be single-stranded for more than an instant. Right away, that new DNA strand will be produced. But what's important to see is that if this is the position where replication began, it seems to be progressing on both sides of the origin. So to define bidirectional, we have to use the word origin. With respect to the origin, DNA replication is bidirectional. It is going to proceed in both directions, and naturally transcription won't. So we're going to have to be ready to make that compare and contrast when we get to that point. From your book, they're showing a cartoon, a sketch to interpret a series of electron micrographs that show this process of bidirectional DNA replication. They're showing an early picture. You can see where replication has begun here and here. And a short time later, we can see that that bubble has grown bigger, that bubble has grown bigger, as though the process of replication is proceeding both to the left and to the right of that original origin of replication. And the replication will continue until all of these replication bubbles kind of coalesce and we have completely copied the entire chromosome. Once per cell cycle. That's going to contrast with transcription as well. The third term we often see applied to replication is symmetric. We can look at the very same diagram. As we separated the two template strands of DNA, each of them serves as a template and is used to copy a new strand. So in that respect, this process is symmetric. Both strands of the DNA double helix serve as a template for replication. That's not going to be the case with transcription. The last term is semi-discontinuous. So it takes me a bit of a diagram to explain what we mean by semi-discontinuous. And as we develop that diagram, we'll also introduce the names of some of the enzymes that are involved in this process. So to depict this, I'll imagine I have double-stranded DNA that is undergoing replication, and so I've opened up the double helix at an origin, which must be smack dab in the middle, and I've exposed, at least locally and temporarily, some single-stranded DNA. The first enzyme to function at this replication bubble will in fact be something called primase. What primase makes is